The last section of this chapter is going to explain why these phospholipids are so important in the cell membranes. You may remember from the previous sections that lipoproteins are created because of the fact that we have a polar side of the phospholipids, the amino alcohol, and a nonpolar tail, which consists of the original fatty acids. The fatty acids then are able to interact with the nonpolar compounds in the middle and transport it from one location to the other because the polar surroundings are able to dissolve in water or dissolve in any other aqueous solution such as blood. So you're able to transport things in cells. This is very similar to the way the cell membrane works. Once again, we have the heads on the outsides, but now we have what's called a lipid bilayer because we have two sets of phospholipids, and the nonpolar sides are interacting with each other in the middle because they're able to mix together because they're both nonpolar, whereas along the top and along the bottom forms a protective shield for those. So you're basically covering up the entire cell with these lipid bilayers. The outside is able to mix along with the aqueous solution such as blood on the outside of the cell, while the inside is able to interact with the cytoplasm and things on the inside of the cell. So both sides need to have polar halves to interact with aqueous solutions. But the center part gives it the ability to hold together and to also bind with nonpolar things like proteins. These are called the hydrophilic region that are the heads. Hydrophilic, if we break down that word, hydro is water, philic is loving or attraction. So hydrophilic are polar heads that are attracted to water. Hydrophobic is the nonpolar tails. Once again, hydro means water, phobic means fear or scared of. So the nonpolar tails want to be as far away from the water as possible, so they are scared of water. So just to sum up again, cell membranes separate what's inside the cell from the external environment. So they're keeping the contents of the cell together. Consists of this lipid bilayer. Those are the phospholipids in two rows where the nonpolar tails are facing each other and the polar heads are facing out. The inner portion are the nonpolar tails, while the outer portion are the polar heads that are then interacting with the aqueous solutions on either side. That bilayer contains proteins, carbs, and cholesterols. So that bilayer can actually interact with all these nonpolar compounds because of those nonpolar tails. Because of the fatty acids and because of the fact that they are not officially attached to one another, that means they can move along the sides of one another, which makes them very fluid, which means that they can rearrange their shapes. They communicate with hormones and neurotransmitters by using the proteins and carbohydrates. These carbohydrate side chains gives it the ability to interact with other things around it, like those hormones. You'll notice also there's little holes in here. That's allowing things to move in and out of the cell membrane. There's 3 different ways that things can move in and out. Diffusion works similar to osmosis, where it's going to go from higher concentration to lower concentration. So if something is really thick, it's going to try to thin it out by getting rid of some of it. Facilitated transport will still go from high to low concentration, but it might need a little help. It increases the rate of that diffusion. And then active transport is actually the opposite of that high to low. It actually goes from a lower concentration up to a higher, but it has to be forced out. So active forces things out. So here's passive, the diffusion. It just does it naturally. Facilitated, it does it naturally, but it has a little bit of help. Whereas active actually goes against the gradient. In other words, here's a low concentration. It's going up to a higher concentration area, which is unnatural, so it needs to be pushed out. So let's take a look. The transport of particles across cell membrane from high concentration to low concentration, so we're decreasing the concentration, it's called diffusion. Does it naturally without any help. That is the end of chapter 15.